Hello, everyone, and welcome back. Sorry, I kind of came in there with a blast today. Today, what our objective is to get through the neuromuscular junction. We may get a little bit further in the lecture today, but I don't want to overwhelm you all. What we're basically going to do is that we kind of told the middle of the story about what happens with muscle contraction and how the actin and the myosin filaments, it's their interaction that allows for contraction to take place. We also discussed the role of calcium, that calcium that's released from the T-tubules that was once stored in the sarcoplasmic reticulum, in the terminal cisternae of the sarcoplasmic reticulum. That calcium is going to leave out, bind to troponin, cause tropomyosin to roll off of the G-actin, F-actin, G-protein, exposing its active site. The myosin head is attached to that active site, and it pulls that thin filament in towards the center. So we really just kind of talked about the middle of what happens in muscle contraction, and today, by the end of the lecture today, we're going to have a very good understanding of what happens at the beginning, what initiates this skeletal muscle contraction. We may get a little bit further in these notes, but as I said before, I don't want to overwhelm you. So keep in mind that we've already discussed how skeletal muscle is under your voluntary control. So any activity that requires instruction from the central nervous system is going to require neural stimulation. That neural stimulation is going to be in the form of nerves. Sometimes we can have glands or different parts of the body that will do things based on hormonal control, but for the the muscular system, there is a bank of nerves that come there. Now remember, once upon a time, and a few lectures ago, we talked about an action potential, and we said how that action potential got us to change from that negative 70 millivolts to a positive number, and that change was going to allow for the interaction between the actin and the myosin to take place. So that action potential, another way to think about it, is it's an electrical signal, and that electrical signal comes from that neural stimulation. So the electrical signal will travel along the axon. Axons carry information away from the nerve, and then it's going to end once that signal gets to the very end of the nerve. So once we get to the end of the nerve, we call that area the synaptic terminal. That synaptic terminal has a couple of important parts to it. First and foremost, that synaptic terminal is going to be right at the edge of the area between the nerve and the muscle, which we call the synaptic cleft. And at the synaptic terminal, this is where we're going to release our communicative device that will tell the muscle to contract. It will tell the muscle to change its resting potential. It will tell the muscle to generate an action potential. And remember, your cells, they don't Facebook, they don't text, they don't tweet, they don't call people on the phone, they don't send letters. So the way that your cells have to communicate with one another are through chemicals. And the chemical that we're going to use as our mode of communication at the muscle junction is a neurotransmitter called acetylcholine, or ACH. Acetylcholine is going to bind to the receiving cell, in this case the muscle, in order to generate an action potential or a change in voltage on the inside of the muscle cell. So a very important thing that's happening here is that at the synaptic terminal, once that electrical activity gets to the end of the axon, the end of the nerve releases at the synaptic terminal of the nerve this neurotransmitter acetylcholine. And that acetylcholine will bind to the motor end plates of the muscle. So here's a beautiful picture of all this happening here. So we have our motor neuron here. That motor neuron comes from your central nervous system, brain and spinal cord. You just told your arm to move up. So when you told your arm to move up, that electrical signal was sent along the length of the axon. And this is a myelinated axon. We'll talk about the importance of myelination a little bit later on this semester. So once we get this action potential down to the end of the axon, which is called the synaptic terminal again, we will have these little red balls on the inside are going to be released. What do you think those little red balls are? Absolutely right. That's our communicative device. That's our acetylcholine, that neurotransmitter. This space, kind of a brownish area between the blue thing, your nerve, and the muscle is what we call the synaptic cleft. And across that synaptic cleft is going to be where these little red balls, acetylcholine, are released. So if we get a little bit closer to that and we see what's going on right there in that synaptic cleft, we see that as that action potential comes to the end of the presynaptic cell, in this case the presynaptic cell is the neuron, so as that action potential arrives at the end of the neuron, these vesicles of acetylcholine are going to be released and they're going to travel across the synaptic cleft. As they travel across the synaptic cleft, they're going to bind to these receptors on the motor end plate of the neuron. So right now, at this very moment, what we're looking at, the potential of that muscle is negative 70 millivolts. Nothing has happened yet. We're still waiting for acetylcholine to bind to these receptors in order for it to generate an action potential in the muscle cell. However, on this nerve, the potential of this nerve, if we were to measure the voltage of it, would probably be at plus 30. So it's gone through this bout of what we call depolarization. So it is generating an action potential. However, we haven't been able to feel or to perceive that action potential in the muscle yet. So the action potential has caused the release of the red balls, the acetylcholine, across the synaptic cleft. And as they are released across the synaptic cleft, these red balls are going to be attracted to the receptors on the motor end plate. The motor end plate is found on the muscle. So the receptors on the muscle is another way to say that. So when those balls attached to the receptors on the muscles, it's going to allow for sodium ions to come in. Notice the charge of those sodium ions. The sodium ions have a positive charge. So let's do a quick recap. When a muscle cell is resting, it's at negative 70 millivolts. Now imagine we're adding all of these positively charged sodium ions. What do you think is going to happen to that negative number? Absolutely right. It's going to go up. And what did we discuss happens when that negative number, that negative 70 millivolts, goes up to a positive number? That generates a action potential. Action potential. Yay! So the influx of the sodium ions is what causes that negative 70 number to increase from a negative number to a positive number. The movement of that negative number to the positive number, that's what we call an action potential. So it's not the acetylcholine that comes into the muscle cell. The acetylcholine is just the lock, the key to the lock that allows sodium to come in. So once acetylcholine binds to those receptors, it allows for sodium ions to come into this resting muscle cell. When those sodium ions come into this resting muscle cell, those positively charged ions come into this resting muscle cell, it takes us from a negative 70 millivolts to a plus 30 millivolts. Going from negative 30 to plus 30 means that we now have an action potential that's been generated. So acetylcholine, or ACH, travels across the synaptic cleft. It binds to the receptors, membrane receptors on the sarcolemma. Remember, sarcolemma is just a fancy way of saying plasma membrane of a muscle cell. And we can also call that area where the acetylcholine binds to the muscle cell the motor end plate. As a result of the binding of the acetylcholine to these receptors on the motor end plate, aka the sarcolemma, aka the plasma membrane of the muscle cell, it's going to cause for sodium ions to come into this sarcoplasm. And remember, sarcoplasm is just a fancy way of saying the cytoplasm of the cell. So when sodium ions come into the cell, it allows for that negative number, negative 70 millivolts for a resting cell, to now go to a positive number. Now we don't want to have all of this sodium ion just coming in all the time, because we don't want this muscle constantly contracting. We don't want it constantly generating an action potential. So what happens is that in the synaptic cleft, all of that acetylcholine is going to be broken down. And it gets broken down by a special enzyme called acetylcholine esterase. Word to the wise. Anything that ends in ASE in biology is typically an enzyme. So acetylcholine esterase is a specific enzyme that is used to break down the leftover acetylcholine that's in the synaptic cleft of a neuromuscular junction, because we might not want, or definitely we don't want, constant muscle contractions to be taking place. So here we see that the acetylcholine esterase is represented by the little moon-shaped jobbies there. It's going to break down acetylcholine into a choline and acetyl, and when that happens, the presynaptic cleft will take up that broken down acetylcholine and recycle it for the next time another action potential comes to this presynaptic cell, and we can release more acetylcholine to bind to the receptor on the motor end plate of the muscle. So that action potential that we've been talking about, what caused us to go from a resting potential, negative 70 millivolts, to a positive number, or to generate that action potential, happened directly as a result of this element because of sodium ions. So because of these sodium ions coming into the cell, that's what triggered the excitation of the membrane. That's what triggered the cell to change from being resting to doing something active. Now, this action potential doesn't just stay in that one area. In fact, it's propagated all along the length of the muscle cell. And one of the jobs of the T-tubules was to make sure that that action potential went to all parts of the muscle cell. And we talked about that on our tour, where we said the T-tubules, their job is to propagate the action potential. So they're spreading that action potential, that negative, going from a negative number to a positive number. They spread that to all parts of the muscle cell or muscle fibers so that the muscle cell contracts together. So that leads to the excitation-contraction coupling. The action potential reaches the triad, which consists of 2 terminal cisternae of the sarcoplasmic reticulum and 1 T-tubule. Once that electrical signal that says, hey, do something, it's going to trigger the triad to release calcium. When that calcium is released, it's going to trigger a contraction. Now, we all know that where it says triggering a contraction, there's a lot that happens in that part, right? Because we understand that when calcium is released, it binds to troponin, causes tropomyosin to roll over, expose the active sites on the G protein, F actin, G actin, whatever you want to call it, and the myosin head is attracted to that exposed active site, and the myosin head pulls that thin filament in towards the M line or the center of the sarcomere, and then ATP will recock that myosin head. So we understand that triggering contraction means a lot more than just triggering contraction. That we know exactly how those actin and myosin filaments are going to interact with one another. So we have a very good understanding of what that means. So just to abridge it, we just say, triggering a contraction. That myosin head is loaded by ATP for energy, and we recock that myosin head and put it back into position for the next time another action potential comes and another more calcium molecules are released from the T-tubules of the triad, and they bind to troponin that cause tropomyosin to roll over and expose the active sites on the G protein, and then the myosin head is attracted to the active sites of that G protein, pulls that thin filament in towards the center, and then ATP comes and recocks that myosin head. So here's an up-close cross-section of that sarcoplasmic reticulum when calcium is released. And what triggers the release of calcium again? The arrival of the action potential through the T-tubules. When that calcium is released, we have our myosin head, we have our active site on our G protein, or G actin, F actin. We have our nebulin, which kind of stabilizes it all together. And then we have our tropomyosin, which is a little green dot there. When calcium is released, it's going to bind to troponin, and when it binds to troponin, it causes tropomyosin to roll off of the active site, and because that active site is now exposed, the myosin heads are attracted towards that active site, and then will pull that thin filament in towards the center. And remember, there are 4 parts that make up a thin filament, all of which we're looking at in this picture. We have nebulin, we have our G actin, aka F actin, aka G protein. We have our tropomyosin, the green dot, and then we have our troponin, which are our little, looks like little brown jelly beans. So that contraction cycle, once again, we were looking at a cross-section on this slide here, but here we're kind of looking at everything up front. Once again, we have our troponin, the brown jelly beans on top of the tropomyosin, the green strands, and we have our F actin, G protein, and the yellow parts are the active sites. Now, there's been no calcium that's released, so everybody's kind of staying in place, and then we have our myosin head here. Once calcium, and I'm going to go back to this page, oops, sorry. I thought I had something else there. So once calcium is released, then it's going to bind to troponin and cause tropomyosin to roll off of that active site. When it rolls off of the active site, the myosin head will be attracted towards the yellow part of the active site of this G protein, and those myosin heads will pull that thin filament, this whole thing, in towards the center of the M line and give us a contraction, in effect shortening our sarcomere. So as that sarcomere shortens, the muscles pull together and that produces a tension. Now, how long that contraction happens is going to depend on a couple of things. One, it's going to depend on how long that neurostimulus is there. So how long or how much acetylcholine was released and left over in the synaptic cleft that allowed for how much sodium ions to come into the cell. So that's going to depend on how long this contraction takes place. How much calcium ions are available, because calcium has to bind to troponin in order for tropomyosin to roll off of the active site in order for it to be exposed for the myosin head to attach to that active site and pull that thin filament in towards the center. So that's going to also have a role to play in how long this contraction takes place. And then keep in mind, every time we pull the thin filament in towards the center, we have to recock that myosin head. And the only way that we can recock that myosin head is with energy, or ATP. So how much ATP is available is also going to have a role in how long this contraction takes place. So from this point on is where you're going to go to your animations. I have tried this several times to get the animations to play over a collaborate session, and they don't really play over a collaborate session very well. Either you can see it, or you can't hear it, or you can hear it and you can't see it. So instead, what we're going to do is that I'm going to show you where these animations are. Just as soon as I can. Well, that's not what I wanted. All right, so if we go to our lecture lab content, and we go to our lecture notes, and then we're going to go down to muscle notes and animations. And these are the animations that I want you to know. So all of these animations that are in here, these are the ones that you should be watching. So when it says go to animations, be sure to view these animations. Everything that we've talked about at the neuromuscular junction, where an action potential travels along the length of an axon of a nerve, and it ends at the synaptic terminal, that action potential is going to trigger the release of acetylcholine. And another thing that helps to allow acetylcholine to be released is that calcium is going to come into that nerve as well. So calcium is going to come into the nerve to trigger the release of acetylcholine. Let's pause real quick. We talked about how calcium is required for bone. It helps to make that hydroxyapatite. We've talked about how calcium is important for muscle contractions. It binds to troponin and causes tropomyosin to roll over to expose the active sites in the G protein, effectively having myosin pull that thin filament in towards the center. And now we're talking about the role of calcium in the nervous system. So calcium comes into the nerve. Once the action potential arrives there, it activates those pumps to bring calcium in, and when calcium comes in, it triggers the release of acetylcholine. When acetylcholine is released, it's going to bind to the motor end plate of the muscle cell, and as it binds to those receptors, it opens the channels for sodium. When sodium ions come into the muscle cell, it creates an action potential that's propagated along the length of the entire muscle cell, which in turn triggers the sarcoplasmic reticulum to release calcium through the T tubules, and calcium is going to bind to troponin, causing tropomyosin to roll off of the active site to the G protein, where the myosin head is now attracted to the active site of that G protein and pulls that thin filament into the center, effectively shortening the sarcomere, and ATP comes along to recock that myosin head. That calcium is going to be actively taken up back by the sarcoplasmic reticulum, where it will be stored. So notice, we have talked about the beginning of a skeletal muscle contraction at the neuromuscular junction with the release of acetylcholine, which is triggered by the action potential and the calcium ions coming into the cell to release the acetylcholine, and the fact that the acetylcholine binds to those receptors in the motor end plate of the muscle cell, and how that generates an action potential in the muscle cell, to what happens in the middle. which is that action potential is going to trigger the sarcoplasmic reticulum to release calcium. Then calcium is going to bind to troponin, causing tropomyosin to roll over and expose the active site on that G protein. The myosin head attaches to the active site on that G protein, and it pulls that thin filament in towards the center. ATP comes, so now we're talking about the end of it, recocks that myosin head, puts it back into place. That calcium is going to be actively taken back into the sarcoplasmic reticulum, way back in the synaptic cleft, where in the neuromuscular junction, where the nerve began to communicate with the muscle. At that point there, we have that enzyme, acetylcholine esterase, breaking down the acetylcholine so that the broken down parts of acetylcholine can go back into the presynaptic cell or the nerve cell to start this process all over again. So all of the parts of skeletal muscle contraction, everything that's going to be on this list, steps involved in skeletal muscle contraction, are found, we've talked about already, and they're also found in these animations. So when we shorten the sarcomere, this is what happens. We have a shortening of the sarcomere, so each one of these little guys, there's a sarcomere, there's a sarcomere, there's a sarcomere, that's going to create tension. And that tension that we've created is going to allow work to take place or to happen. Now, the end of it, we kind of talked about this just a second ago, where acetylcholine esterase breaks down the acetylcholine in the synaptic cleft of the neuromuscular junction, and how ATP is recocks that myosin head, and it's broken down into ADP and inorganic phosphate. And we even talked about how calcium is going to be actively taken back into the the sarcoplasmic reticulum. So specifically the terminal cisternae of the sarcoplasmic reticulum. So all of these things here we've kind of already talked about. Calcium concentrations are going to fall because the calcium is actively taken back into the sarcoplasmic reticulum. Calcium detaches from troponin. The active sites are recovered by tropomyosin because there's no longer any calcium attached to the troponin. So if there's no calcium attached to the troponin, then tropomyosin can go back to its normal resting position. And at this point here, since there aren't any myosin heads pulling that thin filament in towards the center, the myosin heads have been recocked, then the sarcomere is going to go back to its resting stage after the myosin head gets uncocked from it. So as long as we have the thin filaments being pulled in towards the center and the myosin head hasn't been recocked back, and remember, what do we need to recock that myosin head back into its normal position? ATP, we need energy. So as long as there is no energy to recock that myosin head, then the sarcomere is going to remain contracted because that myosin head is going to still be pulling in towards the thin filament in towards that end line. What rigor mortis is the fixed muscular contraction that takes place after death. What allows for this process to take place while we have these fixed muscular contractions very shortly after death is because your body no longer is making the energy or ATP needed to recock the myosin head, nor is it making the energy or ATP that's responsible for pumping those calcium ions back into the sarcoplasmic reticulum. So the calcium is going to remain bound to the troponin, allowing that active site to remain exposed longer because tropomyosin has rolled off of that active site, and the myosin filament continues to pull or keep that thin filament pulled in towards the center. So we have this unavailability of ATP because you're dead and you're not making any energy because you're dead. And then we also have this buildup of calcium because there isn't any ATP that will allow the pumps to work that usually pump calcium out of the sarcoplasm or the cytoplasm of the muscle cell back into the sarcoplasmic reticulums. Depending on the amount of rigor mortis that is set in, investigators, scientists are able to tell within hours and sometimes minutes of when this person expired, because eventually the calcium will sort of diffuse out of the area and it will allow the body to be flaccid. So here is a recap of the events of the neuromuscular junction, or the whole process of contraction from beginning to end. So let's go. The action potential is going to arrive at the synaptic terminal of the presynaptic cell or the nerve cell. Calcium is going to come in, and as calcium comes into the nerve cell, it's going to cause acetylcholine to be released. When acetylcholine is released, it will bind to the receptors on the motor end plate of the muscle cell, triggering another action potential. When that triggering of the other action potential takes place, it will reach the T-tubule, which will then cause the sarcoplasmic reticulum to release calcium. When that calcium is released, it's going to bind to troponin on the thin filament, causing tropomyosin to roll over and expose the active sites. Those active sites being exposed is what allows for the cross-bridge formation to take place. The myosin is attracted to those exposed active sites. When myosin attaches to those exposed active sites, there will be a pivoting motion of that myosin filament that pulls the thin filament in towards the center, effectively shortening the sarcomere and causing a contraction. After that contraction, we're going to have the acetylcholine that was left over in the synaptic cleft of the neuromuscular junction be broken down by the enzyme acetylcholine esterase. ATP will allow for the calcium channels to actively pump back up the calcium into the sarcoplasmic reticulum. Because the active sites are no longer exposed, because calcium is no longer bound to troponin, so tropomyosin can roll back on top of the active site, the myosin head, with the help of ATP, will be recocked back into position. Because the contraction is ended and there's no longer an interaction between the myosin head and the active site of the G protein, the contraction ends and the sarcomere goes back to its resting length. Ta-da! Wasn't that fantastic? And isn't that amazing? Because keep in mind, we're talking about things as small as sodium ions, calcium ions, proteins. This is all stuff on a molecular level. So this is all the physiology. All this stuff is happening just as you move your arm, as you scratch yourself, that you're having this whole elaborate process taking place in your body all the time. Isn't that fascinating? Well, it is for me. So the next little bit that we're going to go over, since we got through that in relatively decent time here, is that we're going to talk about skeletal muscle tension. So now we're going to sort of look at the big picture of all of this. We're not just going to talk about what's happening to an individual muscle cell. We're going to see what happens to this group of muscle cells. So we're taking the microscope from just focusing on one muscle cell and how it contracts to what does this mean for an entire organ of a muscle. So we'll talk about muscle tension. We're still in chapter 10, and even if you're in the new textbooks, you should be kind of looking over into the next section. It should flow nice and organically into the muscle tension segment of the note or of your book. So muscle tension it follows the all or none principle. So that means you don't get one part of a muscle cell contracting and the other part doesn't, because that action potential, once again, is propagated along the entire length of the muscle cell. What happens in the north end of the fiber is going to be what happens in the south end of the fiber. So as a whole, a muscle fiber is either contracted or it's relaxed. Either it has an action potential or it didn't. You don't kind of have any half action potentials that take place there as a general rule. The tension of a single muscle cell is going to depend on a couple of things. First of all, it's going to depend on how many cross bridges that were formed. How many places did myosin interact with the active site on the G protein or F-actin or G-actin? How many times or how many places did that happen? It's also going to determine on what was the resting length at the time of stimulation. Any sarcomere where the resting length is too long is not going to form enough cross bridges, so you're not going to get a nice, strong contraction. Any place where there are too much overlap between the actin and the myosin filaments, neither is that going to give you a very strong contraction on there either, because there's too much overlap. And there's, in fact, I'm going to show you all a laboratory with a frog muscle that kind of shows that exact same thing. So I'll show that to you all in just a second. And then it also depends on the frequency of the stimulation. How many times did we have a neurotransmitter, that acetylcholine, get released at that synaptic terminal? How many times did that happen? So this tension of that muscle fiber has got a couple of things that it's dependent upon when we talk about the strength of that tension or how much force was been exerted. So that length-tension relationship, one of those factors that depends upon which the tension of a muscle fiber is dependent. The number of pivoting cross bridges depends on how much overlap is between the thin and thick filaments. The maximum overlap is going to give you the greatest amount of tension, where the optimum overlap gives you the greatest amount of tension, where we have just the right number of thin filaments for thick filament portions of it. If you have too much or too little, then your muscle cell is not going to be nearly as effective as we would like it to be. So I'm going to find that physiology dynamics I wanted to show you all. And I'm going to make this nice and large, as large as I can make it. I'm going to go back and I'm going to share my desktop with you. That's not what I wanted. All right, so notice when we have our muscle length here at 75 millimeters, and when we stimulate this, we get a nice chart or trace here. We get a beautiful trace. And what that trace is showing us is a couple of things. We're at resting potential here. Notice that we're not looking at levels of force at this point in time, but things that this trace will tell us is that before the contraction, we're at resting potential. As we generate more force, this could be an example of the action potential, and then we're going to come back down to resting. So it's really showing us all of the parts that we know, or all the things that we know that they're going to happen in a muscle contraction. It goes from negative 70 millivolts to a positive number, and then after the contraction it goes down. Now notice what happens if I increase, oops, I forgot to record the data. Okay. So if I increase my muscle length. So I've increased it to the maximum muscle length. Keep in mind I've stretched it out. So you've watched that process take place. We've stretched out that muscle cells, muscle fibers. When I do that, let's look to see what the active force is going to be here, or active force and the total force. So we'll stimulate and record that data itself there, that for our active force here, we only have 0.11 for our active force. We have a lot of passive force that's taking place over here, but we don't have nearly as much active force that is taking place because we have allowed those zones of overlap are too far apart from one another. So they're not, the myosin heads aren't able to actively pull in the thin filaments towards the inline. So we're really just looking at the force that's been exerted passively through the activity of the tendons. Now what happens if I make this muscle as short as I possibly can? And we're going to stimulate, record our data on there. Notice how these two numbers are the same for our active force when the muscle was too long and when the muscle was too short. So that just goes to show you that the optimal length of it was where it was at, I think, about 75. When that muscle was at its optimal length, we were able to generate the most force. So our active force was generated more when we were at the nice length where we had the appropriate number of thin filaments interacting with thin filaments. If there's too much overlap of them, if they're too close together and the muscle's too short, or they're too far apart, the muscle's too long, neither one of those are actively going to give us a nice, good contraction. So here's a pretty picture of all the things that we just looked at on the previous, on that animation that we just worked with, that physio lab. So when we have the sarcomere is too short, we don't get a whole lot of good efficiency there. It's very low. If it's too far apart, it's still very low. The optimal range here is where these are thin filaments, these are our thick filaments, where we have the appropriate level of overlap. Not too much, not too little. Other things that are going to dictate what's the tension that's produced here is the frequency of stimulation. So a single neural stimulation produces what we call a twitch, and it can last for something as short as 7 milliseconds or something as long as 100 milliseconds on there. And there's such a large gap between how long it's going to be simply because we're talking about different types of muscles. So the soleus muscle, which is that deep muscle in your calf there behind your gastrocnemius, that muscle is going to have a much longer twitch than something like a muscle on your eyelid that's going to have a faster twitch then. To sustain these muscular contractions, we're going to need repeated stimuli. We're going to need lots of acetylcholine that continuously binds to the receptors on the motor end plate and allows for sodium ions to come in to generate an action potential on the muscle cell and all the stuff that happens with that. And then the acetylcholine esterase being break down the acetylcholine, and then have another stimulation of more acetylcholine get released, and it's binding to the motor end plate of the muscle cell, allowing for sodium ions to come in, and then it causes for the action potential to take place in the inside of the muscle cell, and then more acetylcholine esterase will break down acetylcholine, and then more acetylcholine comes in. So that whole process that we just went through, in order to remain these sustained contractions, we need lots of stimuli for that to take place. For a twitch, there are 3 phases of that twitch. The latent phase, which is right before the right before the contraction. That's where the active potential is going to move through the sarcolemma and causes calcium to release. The contraction phase is when that calcium actually binds to troponin, causing tropomyosin to roll off of the active sites of the G protein, and that myosin head pulls that thin filament in towards the center. And then the relaxation phase, where those calcium levels fall, they go back to the sarcoplasmic reticulum, the active sites are now recovered, and then the sarcomere goes back to its resting length. So 3 parts of that contraction. And to be honest, we looked at all 3 parts of that contraction when we looked at the frog muscle being pulled apart. So same picture we saw with the frog muscle, same picture here. It's just that now we are labeling this parts of a twitch. And remember, a twitch is a skeletal muscle contraction. We have our latent phase, then we have our active contraction phase, and then we have our relaxation phase. Notice that the time of the stimulation goes from, well, I guess right here, some are as short as maybe 15 seconds, some are as long as about 40 seconds, and then some are up to 100, depending on the muscle. Soleus muscle, that deep calf muscle, very broad, very big, has a very long twitch. Gastrocnemius has a much shorter twitch than the soleus muscle, and then the eye muscles have an even shorter twitch than that. But for all 3 of these different types of muscles, they're all going to have those 3 different phases of latent, contraction, and then relaxation. So here's a picture showing the same thing. It's just that now here we have our stimulus that comes. The acetylcholine is released, binds to the motor end plate of the muscle cell, allows for sodium ions to come in. That action potential triggers the sarcoplasmic reticulum to release calcium. And when calcium binds to troponin and exposes those active sites, boom, we're going to have our contraction phase that takes place. That contraction phase, at that point, at the peak of it, we're going to have the maximum amount of tension that's built. And then from there, we'll have our relaxation phase, where all the stuff that needs for relaxation to take place happens. Acetylcholine is broken down by acetylcholine esterase. The calcium ions are actively taken back into the sarcoplasmic reticulum. Tropomyosin goes back on top of the active site because troponin has regained its normal shape, and then the sarcomere goes back to its resting length. Now, there's a lot of things that we can do with our understanding of muscle tension. And one of the things that we can do or understand about it is how we can have a stepwise increase in tension, so that if you have a stimulation that takes place, and then we have another stimulation that takes place immediately following its rest, the muscle cell rises up and it says, I need to build more tension. And we call that stair-step increase in the twitch tension trepe. So after repeated stimulations, and I'm going to underline this because it's pretty important, after repeated stimulations, immediately after relaxation, you're going to have an increase in the amount of tension that's produced there. So this causes a series of contractions with increasing tension. So if we were to look at this on a graph, what we notice were, and I'm going to come back to that in a second, what we notice would be something that looks like this, that we have the green arrows represent the stimulation, and we are allowed to rest, and then we have another stimulation comes immediately following relaxation. That's the key word. The stimulus has to come following relaxation. And then we have an increase in tension. So the stair-step increase is what we call trepa. With wave summation, that's going to be increasing the amount of tension for every subsequent contraction in much the same way that we saw for trepa. The difference here is that the stimulations come before the end of relaxation. With trepa, we have the stimulations coming right after relaxation. So we still are going to increase the tension or summation of those twitches, but instead of it being a stair step where we go up, down, up, all the way down, up a little bit more, all the way down, notice how we're allowed to relax to go all the way back down. Instead of that taking place, it's going to look more like this. Notice how we never get back to the baseline. We never get a chance to relax. So in wave summation, we have another stimulation that takes place before the end of the contraction. Now, what wave summation can lead to are different types of tetanus. In incomplete tetanus, that's where the twitches are going to reach the maximum tension. If we continue to have this rapid stimulation and the muscle's not allowed to relax, those twitches give you the maximum amount of tension that can be produced there. In complete tetanus, this takes place if the stimulation of the frequency is so high that the muscle never begins to relax, and in fact it is continuously contracted. And the muscle can't hold on to that continuous contraction for too long before it eventually just gives out and it will be forced to rest. So we've already looked at the trepe. This would be the wave summation. Notice that the green arrow, which is the stimulus, comes before the muscle fiber gets a chance to relax. And that wave summation can lead to incomplete tetanus here. So we're having these sustained contractions, but it's not the maximum amount of tension. In complete tetanus, we have this sustained contraction, reaches the maximum amount of tension, and that maximum amount of tension can only be sustained for so long until the muscle just completely gives out. So when we want to develop this tension by the whole skeletal muscle, it's going to have some things that it depends on: how many muscle fibers are stimulated, how much external tension or those passive forces that are exerted on the muscle fiber by those tendons, and then also the internal tension produced by the filament itself, the myosin and actin filaments inside the muscle fiber. That's going to be, it also has a role to play in how much tension is produced in the skeletal muscle. So far, we have really just kind of only talked about individual muscle cells. And when we talk about tension, very rarely are we looking at just individual muscle cells. Even when we looked at the kind of lab simulation of the frog muscle that we just showed here in this lecture, even when we looked at that, we noticed that we were looking at the entire muscle itself, the whole muscle. So tension produced by whole muscle cells happens with motor units. Motor units are going to contain hundreds of muscle fibers on there. That's what makes up the organ of the muscle cell. And one nerve is going to have an effect on multiple muscle fibers. That one nerve plus the multiple muscle fibers that it affects is what we call a motor unit. So because that nerve is releasing acetylcholine at the same time, and those muscle cells are receiving that acetylcholine onto the receptors of the motor plate and allowing sodium to come in to change in action potential, they're all going to have contractions at the same time. So a motor unit is controlled by just one single neuron. One neuron affects hundreds of different muscles there. When we need to pick up something that's heavier maybe than we thought it would be, like if I'm going to pick up my pencil as opposed to picking up the desk, I have to recruit more motor units. So for picking up the pencil, that might only take a couple of motor units. To pick up the desk is going to take many more motor units. And that process of getting additional motor units to fulfill the job that needs to be done is what we call recruitment. So in a whole muscle or a group of muscles, that smooth motion and that increase in the amount of tension is produced by slowly increasing how many motor units are going to be stimulated. Another good example would be you're doing wall sit. So Wall sits are basically you put your back on the wall and then you slide it down until your legs are in about a 90-degree angle, and it looks like you're sitting in a chair, but you're not sitting in a chair. Only your muscles are holding you up. Now, when you first sit in that wall sit, you could probably do 5 or 10 seconds very easily and hold a conversation with someone, depending on how well shape you're in. Now, as you sit on that wall for a little bit longer, or if you're doing planks, same process works. As you sit on that wall a little bit longer, you're going to have to recruit more motor units to come in and hold your body up. So it needs to be able to sustain that tension. Now, if you sit on that wall for too long, even if your mind is saying, continue sitting on the wall, continue sitting on the wall, you can do it, you can do it. After about an hour in, even for the most fit people, they reached maximum tension. So that means all the motor units in those muscles that are required to put you on that wall sit or to have you hold that plank position, those are going to eventually give out. You can only sustain maximum tension for a very short amount of time. And no matter how much your mind says, continue sitting on that wall, continue sitting on that wall, continue sitting on that wall, at some point, they'll just all give out. And we saw that on the previous slide, and I'll go back to show you, to remind you, right here, where we reach that maximum tension, and we can only hold it for so long, and then those muscle cells, even the ones that we've recruited, are going to give out. Because of that recruitment, that's what allows us to develop more tension in either the wave summation or in the trepa. So here's a wonderful picture. The purple neuron is going to affect all the purple cells, so that's one motor unit. The blue neuron affects all the blue cells, it's another motor unit, and the red one affects all the red cells. And all of those muscle cells will contract together. All the muscle cells of that unit will contract one with the other. And here's another good picture of showing that recruitment there, and this must look a lot like wave summation that we have going there, that the more motor units you have working together, the more tension that's going to be produced in this muscle. So sustained tension is less than the maximum tension, and what it allows the motor units to do is to rest in rotation. Muscles that will do this would be like the muscles of your back and of your neck, where those muscles need to hold your neck up above your shoulders for prolonged period of time to sustain those contractions. So instead of having all of those muscles and all those motor units all contracting at the same time for your neck, we're going to let them take turns. So some will be contracted while others are not. So that's what gives us a sustained contraction or sustained tension, and it allows for your head to rest above your neck all throughout the day without it just completely giving out and then your head just drops down to your chin to your chest. Muscle tone is the normal tension and firmness of your muscle even when it's at rest. Your muscle units actively maintain these body positions even without motion. So a good example again would be your neck muscles and also your back muscles, your abdominal muscles. It's maintaining your body position even though they're not actually moving. They are actually working. So as you increase your muscle tone, not only does it look nice, people with nice toned muscles look good, but it also increases your rate of metabolism, because even though those muscles aren't actively having any motion, they're still doing work and having some contractions that will take place there that uses energy, even when they're at rest and not actively moving. So when I say rest here, I mean that they're not having any motion. You're not actively doing crunches there. But even when you're just sitting in the chair, the tone in your back muscles, the tone in your abdominal, all of that's your core, your back, and your front, your ventral and your dorsal, or your anterior and posterior sides of your thoracic and abdominal region, they're still using energy. So nice, sexy toned muscles don't only look good, but they're good for you because it increases your metabolic rate. So we have two types of muscle tension that can be produced, isotonic and isometric. In isotonic contractions, and I'm going to underline this because it's important, in isotonic contractions, the muscle changes length. So as a result, isotonic contractions result in movement. So if the muscle tension is greater than the load, then the muscle is going to shorten. So if the load itself is lighter or less than the tension that can be produced by those muscles, your muscle is going to shorten. Think about this as doing bicep curls. So when you're pulling the weight up closer to your shoulder, then we call that a concentric contraction. The muscle shortens. Conversely, if the muscle tension is less than the load, you don't have nearly as much, you're not actively pulling that weight towards you, you're letting it go back down in the down portion of your bicep curl, that's what we call an eccentric contraction. That's going to lengthen your muscle cell. So in doing bicep curls, that's toggling between concentric and eccentric contractions. They both fall under the category, because the muscle is moving, of an isotonic contraction. So if isotonic contractions are changes in muscle length, then what do you think isometric contractions are? That's right. Skeletal muscle develops tension, but it doesn't actually change any length. That is an isometric contraction. Great example of that would be like carrying groceries. So if you're carrying groceries, you are developing muscle tension in your arms, or another example is doing planks or doing just holding in the push-up position. You're not really changing the length of those muscles, but they're still developing tension. They're still doing work. So isometric contractions, muscles doesn't change any length. However, it's still doing work. Isotonic contractions, you have concentric, where you're reducing the angle of that bond, pulling things up towards you, and eccentric, where you're lengthening it. So concentric shortens it, eccentric lengthens it. Both of them fall into the category of an isotonic contraction. So for resistance and speed, they're inversely related to one another, and this sort of makes sense because the heavier the load on the muscle, the longer it takes for that shortening to begin. The heavier something it is, the longer it takes for you to pick it up, and the less the muscle will shorten because it's very heavy. The lighter the load, the easier you're able to pick it up, the more you can have that muscle shorten. And muscle relaxation, after a contraction, that muscle fiber goes to its resting length by elastic forces of your tendons at the end of those muscles. The opposing muscle contraction, so where your bicep is contracting, your triceps is relaxing, and vice versa, where your biceps is relaxing, your triceps is contracting, and also because of the job and the role of gravity. So here's just a nifty picture that's talking about the inverse relationship between the size of the load and the speed of the contraction. With a large load, notice that it takes the distance is shortened not nearly as much. With a small load, you're going to have a much greater amount of distance that it's shortened. The muscle fibers are shortened. And those elastic forces we talked about, those tendons and those ligaments at the end of it, all of these things help to put those sarcomere back to resting length. And then those opposing muscles like biceps and triceps or quadriceps and your hamstring group back there, they work in opposition of one another, what we call antagonistically, and that also helps to get the muscles to relax, the ones that need to relax. All right, so that is where we're going to end with this session. We got a little bit further than I thought we would get through. On Monday, we'll finish up Chapter 10 because we're going to go through energy for muscle contraction. That's the last little part of it. And if you're taking microbiology, or if you've taken an introductory to biology class very recently, the role of glycolysis and ATP synthesis, all of that may probably be familiar to you, but that's what this last little portion of the notes are. All right, so I hope you have a fantastic day, and if you have any questions, comments, or concerns, don't hesitate to email, and definitely don't hesitate to ask while in class. Have a good one.